Alright, so this is the second lecture of infectious diseases. Right, in the previous lecture, we have looked at the innate immune system. So today, we will be looking at the link between the innate and adaptive immune systems before we then look at the adaptive immune systems. Alright, so for the innate immune system, we have looked at it last lecture. Alright, so I'm not going to repeat myself. Right? I'm just going to leave you to read some of these summarized points about the innate immune system. And next, we'll be looking at the adaptive immune system in the next recording. Right? So for this recording, we'll be looking at the link between the two. How does the innate immune system in turn activates the adaptive immune system? Because the adaptive immune system, as you can read here, requires activation, right? So the activation is carried out by antigen presenting cells. So from the previous lecture, I hope you recall the identities of the antigen presenting cells. All right, so those are the monocytes, macrophages, and dendritic cells, as we have mentioned in the previous lecture. Okay, so what exactly do the antigen presenting cells do, right? So they will recognize the pathogen via their receptors. So remember that the monocytes, macrophages, and dendritic cells are cells of the innate immune system. So they do have receptors that recognize broadly the common antigens, right, found on most of the pathogens. Okay, so the recognition is not specific, but nonetheless, there is recognition, and they will know that there is presence of pathogens. Right, then they would go on to phagocytose the pathogens and digest them. Right, so that is shown here by the eating of the bacterium by this dendritic cell. Okay, so after that, once the pathogen is digested, most of the parts of the pathogens is either absorbed by the antigen presenting cell or released. But some of the fragments, especially the peptide fragments of the antigens found on the pathogens, would be presented or displayed on the cell surface of the antigen presenting cell. And that is with the aid of a protein, a membrane protein called MHC class 2 protein or MHC class 2 molecule. Okay. So once that is presented, right, when the antigen peptide fragment is presented on the surface of the antigen presenting cell, then a naive helper T cell will be able to recognize this presented antigen and will bind to the presented antigen. So that is shown here in this cartoon. Okay. The binding of the naive helper T cell would then activate these helper T cells to carry out its function, and its function would lead to the activation of the adaptive immune system or adaptive immune responses. Okay, so just a bit of explanation for some of the terms. Yeah, so naive here refers to unactivated cells. Okay, you're going to see this word naive later for the T cells and for the B cells as well, right? So because cells of the adaptive immune system needs to be activated, so before they are activated, they are described as naive. Okay, and you will see me, right, using the word lymphocyte in the slide, but I have used the word cells in the notes, and I'll be talking about cells. I will be using the word cells in my own explanation as well, right? So note that the T lymphocytes, B lymphocytes are just T cells and B cells, just different names for the same cells. Okay, so I'm going to use these two words kind of interchangeably. Okay, all right, so that is the whole process of antigen presentation by antigen presenting cells. Okay, so another diagram to show the antigen presentation. All right, so this would be inside of the antigen presenting cell, a macrophage, for example, or a dendritic cell. Right, so again, the phagocytosis as shown here. After that, the digestion of the phagocytosed pathogen. Right, so that would require the fusion of the lysosome, right, to the phagosome, but that is not shown here. Okay, so imagine that there's a lysosome that is fusing with this phagosome, and that brings about the digestion of the pathogen. Right, so all these are the peptide fragments, right, of the antigen on the pathogen. Okay, so these vesicle now, the phagosome with these digested fragments of the antigen would then fuse with another vesicle containing the MHC class 2 molecule, which is a membrane protein. Okay, so the fragment would bind to the MHC class 2 molecule to form a MHC class 2 antigen complex. Right, so this complex would then be inserted into the cell surface membrane of this antigen presenting cell. Right, so now the antigen or the fragment of the antigen is presented or displayed on the cell surface of the antigen presenting cell. All right, so I hope this makes the process a bit clearer, right? Another diagram to show the same thing, right? So this is a dendritic cell with the phagocytosis of pathogens, right? Both here and here, right? After that, the lysosome, as you can see here, lysosome would fuse with the phagosome, right, to digest the pathogen. Then you have the MHC class 2 proteins, like they are not floating in the cytoplasm, yeah, so they should be found in the vesicle, okay, as you have seen from the previous diagram. So anyway, the fragment of the antigen would then bind to the MHC class 2 protein to form this complex, which is then inserted onto the cell surface membrane to be presented, the antigen fragment to be presented to the naive helper T cell here. Okay, all right, so I hope that was clear. Okay, now a bit more on the MHC molecules, right? There are actually two types, right? The class 2 molecules that you have looked at just now. The class 2 molecules are only expressed in the antigen presenting cells, okay? Only expressed in antigen presenting cells such as dendritic cells, macrophages, and monocytes. And there is another kind of MHC molecules known as MHC class 1 molecules, and these are expressed in all our healthy and nucleated cells with nucleus, so not red blood cells, but all the other cells would have MHC class 1 molecules on the cell surface. Okay, so they do roughly similar things. Both MHC class 2 and class 1 molecules present antigens on the cell surface. So we have already looked at how MHC class 2 present the fragments of antigens from pathogens that are phagocytosed, right, on antigen presenting cells, and that would activate the naive helper T cells here. Right, for the MHC class 1, they do not present antigens from phagocytosed pathogens, right? Because not all our cells do phagocytosis. What they present would be antigens that are synthesized inside the cells. So why would there be antigens synthesized inside the cells, right? That would happen if our cells are infected. All right, so when our cells are infected, then as you have learned, say for example viral infection, right, the virus would make use of the infected cell to make viral proteins, right? So some of these viral proteins can then be presented on the cell surface of the infected cells with the MHC class 1 molecules, and that would activate cytotoxic T cells to kill the infected cell. All right. So the recognition of infected cells would be via this antigen presentation using MHC class 1. So all our cells can do that because all healthy nucleated cells have MHC class 1 molecules. So as long as our cells becomes infected, then they can tell the cytotoxic T cells that they are infected and therefore get killed by the cytotoxic T cells to prevent further spreading of the, say, virus, for example. All right, so two types. Okay, but then our focus, right, in this part of the lecture would be on the MHC class 2 because we are talking about antigen presenting cells, right, to link the innate and the adaptive immune systems. Right, but know that there are MHC class 1 molecules as well. Okay. All right, so yep, as we have mentioned, right, the MHC class 2 on the antigen presenting cells would be the one that activates the adaptive immune system by activating the naive helper T cells. Okay, right, so another diagram to basically show the same thing. So I'm not going to repeat myself. I'll just let you maybe look at some of the words in the diagrams yourself. All right. Okay, so that would be the link between the innate and adaptive